Welcome, dear children, to today's session. The topic of the day is digestion in man. We shall look into how our digestive system works. Digestion in man starts with the mouth. The mouth breaks the food down into very small particles, and in the process, the food is mixed up with saliva. Saliva contains salivary amylase, which acts on starch and converts it to sugar. The food that is masticated in this manner is passed through the esophagus from the mouth to the stomach. In the stomach, gastric glands secrete digestive juices. Digestive juices consist of three components: hydrochloric acid, pepsin, and mucus. Hydrochloric acid provides an acidic medium for the action of protein digesting enzyme pepsin. Pepsin digests proteins to peptones. There is also presence of mucus, which will form a uniform coat on the inner lining of the stomach and protect the stomach from acids. Next, the food passes into the small intestine. The small intestine is a long coiled tube and it receives secretions from the liver and the pancreas in its first part. The part is called as duodenum. These digestive juices and the secretions from the liver and the pancreas act on the food to further break it down into usable form. The liver is the largest gland in the body. It secretes bile juice, which is temporarily stored in the gallbladder. The bile juice is involved in breakdown of fats. The pancreas secrete pancreatic juice, which breaks down carbohydrates and proteins. The intestinal juices complete the digestion of starch into glucose, fats into fatty acids and glycerol, and proteins into amino acids. You can have a look at the picture of the system. The food passes from the mouth to the esophagus and to the stomach. On the way to the small intestine, this is the first part of the small intestine, which is called as duodenum. Into the duodenum, the liver secretes bile juice and the pancreas secretes pancreatic juice. So these juices act upon food for the digestion of food. After the food moves in from the first part of small intestine, it passes through the remaining of the small intestine where the food is both digested and absorbed. The last part of the small intestine is involved in absorption of food material. The nutrients are absorbed in usable form in the small intestine and then the remaining matter is passed on to the large intestine. The large intestine has got three parts: the ascending colon, the transverse colon, and the descending colon. In the large intestine, the water is reabsorbed. Water required for the body is reabsorbed. The remaining matter, which is unusable, undigested food, is passed on to the rectum. This part is the rectum, where it is temporarily stored before it is passed on to the anus. Anus is the region which throws out the undigested food out of the system. Let us have a look at how absorption of digested food takes in the small intestine. The digested food is absorbed by the walls of the small intestine. This process is called as absorption. The small intestine has got several finger-like projections which are called as villi. These villi increase the surface area for absorption. The absorbed materials are carried by the blood to different parts of the body and used by the body. This is called as assimilation. The undigested food then passes into the large intestine. In the large intestine, there is reabsorption of water. The remaining Undigested food is converted as feces and then it passes through the anus. The process is called as digestion. Let us now perform an activity to test the effect of saliva on starch. Take two test tubes, test tube A and test tube B. In test tube A, add some boiled rice which is stewed for 3 to 5 minutes. Mix it up with water, shake it and keep it aside. In test tube B, add some boiled rice which is mashed and mixed with water. Now, take 2 to 3 drops of iodine and add to test tube A and test tube B. You will observe that in test tube A, iodine has got its own color, that is golden yellow, whereas in test tube B, the iodine has turned the contents into blue-black color. In test tube A, we have added boiled rice which has been chewed for 3 to 5 minutes and which has got converted to sugar. Salivary amylase present in the saliva converts starch to sugar. So, iodine does not give a positive test in test tube A. Iodine is a reagent which converts starch to blue-black in color, whereas in test tube B, iodine reacts with the starch and turns it to blue-black color. This activity is to be recorded in the classwork with today's date and in the index column written as activity, name of the activity with date. Thank you children for attending today's session.